Somewhere in the North Pacific, thousands of feet below the surface, a shark the length of a transit bus is hunting in complete darkness. It is almost completely blind, and it may be one of the most successful predators the ocean has ever produced. The Pacific sleeper shark is not a small bottom feeder surviving on whatever happens to drift past. National Oceanic and Atmospheric Administration Fisheries has described it as possibly the largest actively hunting fish in the ocean. The largest individuals ever filmed have been estimated at roughly 23 feet long, yet no human has ever physically measured one of those giants. They appear briefly on deep-sea cameras, sometimes thousands of feet below the surface, and then disappear. But their size is only the beginning. Scientists examining Pacific sleeper shark stomachs have found harbor seals, stellar sea lions, giant Pacific octopus, adult Chinook salmon, and other powerful animals. Some of that prey can move many times faster than the shark hunting it. The sleeper shark cruises at less than half a mile per hour. It cannot see its prey. It catches them anyway, and the reason for its blindness makes the contradiction even stranger. Nearly every Pacific sleeper shark examined in some Alaskan studies has carried evidence of a parasitic copepod attached directly to the eye. The parasite anchors into the cornea, damaging and clouding the tissue until vision can become effectively useless. Both eyes can be affected, yet the shark continues feeding. Something else is guiding it through the darkness, and there are other parts of its life that remain almost completely hidden from science. No pregnant Pacific sleeper shark has ever been documented. Researchers still do not know where these enormous animals give birth. Even their growth unfolds on a timescale difficult to imagine. One large female studied in the Aleutian Islands was already decades old and had apparently not reached sexual maturity. An animal this large can spend much of a human lifetime simply growing up. In today's journey, we are following this shark into the cold depths where speed matters less, eyesight can become almost irrelevant, and a predator moving barely faster than a drifting current can somehow capture animals built to escape. Long before forests cast their first shadows across dry land, sharks were already moving through the seas. Their ancestry reaches back more than 400 million years, beyond the first trees and far beyond the first dinosaurs. Across that immense span, oceans opened and closed, continents shifted, climates swung between warmth and ice, and entire branches of life vanished. Sharks remained, diversifying into forms suited to reefs, coastlines, open water, and eventually the cold depths where sunlight becomes irrelevant. Among those branches is the family Somniosidae, the sleeper sharks. Its name traces to the Latin word somnus, meaning sleep, a reference to the sluggish appearance that early observers associated with these animals. The label sounds almost gentle, yet it captures only what humans noticed from brief encounters. Sleeper sharks belong to a lineage shaped around low energy expenditure, cold water, slow metabolism, and extraordinary longevity. Their bodies favor efficiency rather than acceleration. They do not embody the familiar design of a predator built to spend energy rapidly and recover it through frequent feeding. This family includes several deep water species, but one member became particularly associated with the northern Pacific. Somniosus pacificus, the Pacific sleeper shark, entered formal scientific literature under that name in 1944, when Henry Bigelow and William Schroeder described the species. Classification gave it a place in zoology, but not prominence. For decades, it remained an obscure presence in fisheries records and occasional scientific collections, known largely through animals encountered by chance. Its geography seemed broad enough even then. Records placed it through temperate and subarctic waters from Japan, across the Bering Sea and Alaska, and southward along western North America toward Baja California. That map appeared to describe an animal tied closely to colder northern waters. Yet maps of deep ocean species often reveal the limits of observation as much as the limits of the animals themselves. In 2025, cameras recorded Pacific sleeper sharks in the South China Sea, far south of the range traditionally associated with them. A species described more than 80 years earlier was still capable of appearing where established expectations had not placed it. Its closest famous comparison is the Greenland shark, Somneosis microcephalus. Both belong to Somneosidae, but they occupy different oceans and follow different biological clocks. Greenland sharks inhabit the North Atlantic and Arctic and have produced age estimates approaching 4 centuries, giving them the greatest confirmed longevity known among vertebrates. Evidence from Pacific sleeper sharks indicates faster growth, perhaps around twice the rate observed in their Greenland relatives. That difference may shorten the Pacific species' lifespan considerably, although its life may still extend across centuries. Shared ancestry has produced 2 enormous cold-water predators whose lives unfold at remarkably different rates. Compared with great white, tiger, or bull sharks, the contrast reaches deeper than swimming speed. Those familiar hunters evolved in environments where rapid movement and visual information can carry enormous value. Sleeper sharks belong to another ecological tradition. Energy is conserved, movement is measured. Their sensory biology is suited to water, where illumination fades and other forms of information become more dependable than an image formed by an eye. Seen within that ancestry, the Pacific sleeper shark becomes less an evolutionary oddity than a highly specialized expression of an ancient design. Its heavy body, damaged eyes, measured movements, unusual prey, and extraordinary endurance belong to the same underlying history. What appears strange from the surface becomes coherent when placed inside a lineage shaped for cold water, scarce energy, immense depth, and time measured on a scale far longer than a human life, across countless generations of survival. A fishing vessel can place a Pacific sleeper shark on a deck, stretch a measuring tape beside it, and still reveal only the smaller end of the species. The individuals physically recovered from Alaskan waters commonly reach roughly 12 to 14 feet, with large specimens weighing from around 700 pounds to nearly 2,000. Beside a human observer, even these sharks occupy an imposing amount of space. Yet the measurements carry an unusual limitation. The animals available to science appear to be juveniles or subadults. That distinction leaves the true dimensions of the species unresolved. A shark 14 feet long can already rival the length of a small car placed end to end with another, yet evidence suggests substantial growth may remain ahead of it. Mature adults seem to spend much of their lives beyond the depths routinely sampled by fisheries, leaving researchers with bodies large enough to seem definitive while representing only an earlier stage of development. One of the clearest glimpses of what adulthood might produce came in September 1989 in Suruga Bay, Japan, from a submersible operating around 4,000 feet below the surface. Observers recorded a female Pacific sleeper shark whose length was estimated at approximately 23 feet. The estimate was possible because the animal passed near structures whose dimensions were known, giving researchers a reference against which her body could be scaled. Another encounter widened the uncertainty further. Off Oahu, Hawaii, a remotely operated camera recorded an enormous sleeper shark at roughly 6,300 feet. Comparisons with objects visible in the footage produced an estimated length somewhere between 16 and 26 feet. The range is broad because estimating an animal through underwater imagery introduces perspective and positioning uncertainties, but the observation still points toward dimensions far beyond most specimens brought aboard fishing vessels. This is why the language used by National Oceanic and Atmospheric Administration fisheries is unusually striking. The agency identifies the Pacific sleeper shark as Alaska's largest shark and has described it as possibly the largest predatory fish in the ocean. The distinction matters. Whale sharks can exceed the sleeper shark in length, as can basking sharks, but both obtain food primarily by filtering small organisms from water. The Pacific sleeper shark belongs to another category entirely. Its size is attached to an animal that actively consumes substantial prey. At 23 feet, one of these sharks would extend farther than the width of a standard school bus. Its mass could exceed that of a large adult male polar bear, while its thick cylindrical body would make the scale more apparent than length alone suggests. Yet no specimen of that magnitude has ever been hauled into a research station and measured from snout to tail. Other giant sharks offer a revealing contrast. Great white sharks have been measured directly, photographed repeatedly, tagged, tracked, and compared across decades of observation. Whale sharks have likewise been measured in open water using increasingly precise photographic methods. For the largest Pacific sleeper sharks, science possesses fleeting images, scale references, and encounters separated by years and thousands of miles. The missing measurement is therefore more than an empty line in a species description. Researchers trying to determine growth, reproductive maturity, feeding requirements, and population structure must work from animals that may represent only part of the species' lifespan. The mature body remains largely outside the physical record. Somewhere beneath the Pacific, the upper boundary of this predator's size still exists as a living measurement no human has taken. Its greatest dimensions remain recorded not by hands or scales, but by cameras briefly sharing the water with animals that soon passed beyond view. Seen at close range, the Pacific sleeper shark seems assembled according to rules, unlike those governing faster sharks. Its skin is dark gray, brownish gray, or nearly black, with little of the bold patterning found on many shallow water species. Against dim, deep water, that subdued surface offers little visual contrast. There are no bright markings to announce the outline, only a broad body whose appearance changes remarkably little from head to tail. The shape reinforces that impression. A blunt, rounded snout leads into a heavy, cylindrical trunk. The pectoral fins are relatively small, while two low dorsal fins sit far back along the body, and neither carries the defensive spine found in some related sharks. Nothing about this silhouette suggests the narrow, rigid profile associated with rapid acceleration. The Pacific sleeper shark carries its mass differently, favoring a form suited to prolonged movement at low energetic cost. Even its flesh reflects that economy. Researchers handling sleeper sharks have described their musculature as unusually soft and flabby compared with the firmer muscle of faster species. In warmer, brighter waters, such construction might appear disadvantageous. Here, it reduces the amount of expensive muscle tissue that must be maintained. Powerful bursts demand powerful machinery, and powerful machinery consumes energy even when it is not being pushed to its limit. The sleeper shark follows another arrangement. Its ordinary cruising pace can remain below half a mile per hour. At that speed, movement becomes a gradual transfer of an enormous body through the surrounding water rather than a continuous series of forceful accelerations. Each tail stroke can be measured, each correction economical. Energy that another predator might spend covering distance rapidly can remain stored inside the animal. Much of that reserve lies in an extraordinary organ. Like other sharks, the Pacific sleeper shark has no gas-filled swim bladder. Buoyancy is assisted instead by a large, oil-rich liver, an organ that can represent a substantial fraction of total body mass. In an exceptionally large 2,000-pound individual, a liver approaching 30% of body mass would amount to roughly 600 pounds. Its lipids include compounds known as alkyldeacylglycerols, which contribute to the low density of the liver while also representing stored chemical energy. The same structure, therefore, helps support the body in water and carries reserves useful when feeding opportunities are irregular. At the opposite end of this economical system sits a mouth built for decisive contact. The upper teeth are narrow and pointed, useful for securing flesh. Below them, broader teeth form cutting edges capable of slicing as the jaws move against prey. Rather than depending entirely on repeated high-speed strikes, the shark can establish a grip and remove tissue efficiently. Suction generated as the mouth opens may also help pull nearby food inward, particularly when the target is soft-bodied or already within close reach. Across the exterior, dermal denticles form a covering of microscopic tooth-like structures. These scales protect the skin and influence the flow of water across the body, reducing unnecessary resistance as the shark moves. Combined with low swimming speed and restrained tail movement, they belong to a design in which wasted motion offers no advantage. No single feature creates the Pacific sleeper shark's efficiency. The effect comes from accumulation, subdued coloration, compact proportions, inexpensive musculature, buoyant oils, specialized teeth, suction feeding, and skin shaped by water itself. Each saves something, whether energy, movement, or effort. The result is a predator whose anatomy invests remarkably little in pursuit. Its body is arranged around proximity, preserving resources until distance has narrowed enough for feeding to require only a brief, controlled act. There is a small animal whose entire adult existence may unfold on the surface of another animal's eye. Omatocoida elongata is a parasitic copepod, a minute crustacean belonging to the same broad group as countless drifting marine forms and distantly related to crabs and shrimp. In sleeper sharks, it has evolved an unusually intimate habitat. Rather than occupying the skin or gills, the adult female settles directly onto the cornea. Attachment becomes permanent through a specialized structure called a bulla. Produced by the female, this anchoring apparatus penetrates and fixes the parasite to corneal tissue, holding it in place as the shark moves through the water. Male copepods are much smaller and remain more superficially associated. The female bears the deeper connection, embedded where transparent tissue should admit light into the eye. Once established, she may remain there for the rest of her adult life. The damage develops around that point of attachment. Corneal tissue can become lacerated and inflamed. Nearby structures swell. The conjunctiva, the delicate membrane covering exposed portions of the eye, can erode. Most consequentially, the cornea loses its transparency. Scarring and opacity transform a surface designed to transmit light into one that can no longer form a useful visual pathway. In heavily affected animals, the result is functional blindness rather than merely weakened eyesight. Evidence from Prince William Sound in Alaska shows how deeply this relationship is woven into the species. In one examination of Pacific sleeper sharks, 7 of 8 individuals carried active infections. The remaining shark bore corneal scarring consistent with an earlier infestation, broader observations from the region found infection or evidence of previous infection in roughly 97% of examined animals. A condition occurring at that frequency is not an unusual injury affecting unlucky individuals. It approaches a normal feature of life for the population being studied. Nor is the damage necessarily confined to a single side. Parasites can occupy both eyes, leaving neither available as an intact visual backup. For many vertebrate predators, bilateral loss of useful sight would profoundly alter feeding, navigation, and survival. Pacific sleeper sharks present a stranger biological outcome. Individuals carrying severe corneal damage continue to feed, grow, and persist for decades. The parasite has not prevented the species from producing animals of immense size or occupying an enormous geographic range. That persistence creates an evolutionary puzzle. The association between Omatocoida and sleeper sharks appears ancient, likely extending across a substantial portion of their shared evolutionary history. Natural selection does not require parasites to become harmless, but a relationship that consistently destroyed a function essential to survival would impose severe costs on its host. The enduring success of infected sleeper sharks indicates that useful vision carries far less importance in their lives than human intuition might suggest. An intriguing possibility has occasionally appeared in scientific discussion. Some researchers have proposed that microorganisms associated with the copepod could produce bioluminescence, potentially creating a faint lure near the shark's head. Evidence for such a benefit remains uncertain, and the idea has not been established experimentally. The parasite should therefore not be treated as a proven partner or hunting aid. Its clearest documented effect remains injury to the cornea. What survives after that injury is the more revealing fact. Nearly universal eye damage exists alongside continued feeding, growth, movement, and reproduction. The shark's world has not collapsed with the loss of a clear image. Across generations, life has continued with eyes increasingly removed from practical importance, leaving vision as only one channel among a much richer collection of biological information. Vision dominates our understanding of predation largely because sunlight makes vision useful. Near the surface, an eye can resolve distance, movement, shape, and contrast before physical contact occurs. Descend several hundred feet, however, and that advantage begins to diminish. Colors disappear in sequence, outlines weaken, and eventually sunlight contributes almost nothing. For a predator spending much of its existence beyond this illuminated layer, losing useful eyesight removes far less information than it would on land or near the surface. Other channels remain open. The broad nasal openings beneath a shark's snout constantly sample dissolved chemicals carried through seawater. Sharks possess exceptionally developed olfactory systems, capable in some circumstances of detecting certain compounds at extraordinarily low concentrations, though sensitivity varies by substance and species. For a Pacific sleeper shark, smell provides information across distances where neither touch nor electroreception can yet identify a nearby animal. Blood, bodily secretions, waste, and chemicals released during decomposition can form drifting traces shaped by currents. A scent does not provide a photograph. Instead, it creates a changing chemical landscape. As the shark moves through different concentrations, its paired olfactory organs can help distinguish where a trace becomes stronger or weaker. Following that gradient gradually narrows the region in which food may be found. Closer in, another sensory network begins resolving movement. The lateral line extends along the shark's body as a system of canals containing sensitive mechanoreceptor cells. Water displaced by a swimming animal produces pressure fluctuations and low-frequency vibrations. A fish beating its tail alters the surrounding water. A diving seal pushes a moving disturbance through it. These changes reach the lateral line and bend tiny sensory structures within its canals, converting mechanical movement into nerve signals. The result is not vision reconstructed through another organ. It is a different kind of spatial information, one based on motion and pressure. Direction, proximity, and changes in movement can be detected without requiring any light at all. At very close range, the shark possesses an even more unusual sense. Across its snout are tiny pores leading into jelly-filled canals called the ampullae of Lorenzini. At their inner ends sit electroreceptor cells sensitive to weak electrical fields. Living muscles generate electrical activity whenever they contract. Hearts beat through electrical impulses. Respiratory muscles move. Fish working their gills create signals that persist even when their bodies are visually concealed. Electroreception allows sharks to detect some of these faint fields at close range. It is particularly useful during the final moments of feeding, when precise location matters more than identifying a distant shape. Experiments with other shark species have demonstrated that electrical cues can guide strikes even when visual information is unavailable or misleading. Many sharks also protect their eyes during close contact, relying increasingly on non-visual information when prey is nearest. For the Pacific sleeper shark, these sensory systems overlap across distance. Chemical information can reveal the broader presence and direction of something edible. Mechanical disturbances refine the movements occurring nearby. Electrical fields provide intimate biological information once the remaining space has narrowed. No single sense needs to replace an eye, because the task of finding prey has been distributed among several specialized systems. Its restrained swimming adds another advantage to this arrangement. Slow movement produces less turbulence than rapid acceleration, reducing the pressure disturbance sent ahead through the water. The sleeper shark can therefore gather information from movements around it while contributing relatively little disturbance of its own. Blindness, in this setting, does not leave an empty sensory world. The animal remains surrounded by information carried as dissolved molecules, moving water, pressure, vibration, and electricity. What an eye would render as an image, the sleeper shark receives as overlapping physical signals, each becoming useful at a different distance, until the living presence ahead is no longer something that needs to be seen. A tracking tag attached to a Pacific sleeper shark records movement that looks almost like breathing on a graph. The animal rises, descends, rises again, repeating broad curves through the water column for hours. Studies have found individuals spending much of their time between roughly 500 and 1,500 feet, while making regular excursions above 330 feet. Rather than holding one depth, they continually reposition themselves through layers occupied by different prey. The pattern becomes especially revealing after sunset. Many fish, squid, and other marine animals move upward at night to feed in shallower water, creating one of the largest recurring migrations on Earth. Sleeper sharks appear to follow this vertical traffic. Instead of selecting a fast animal and pursuing it across open water, the shark moves with the broader distribution of prey, placing itself where encounters become more likely. Its hunting ground shifts upward as the food web rises through the darkness each night. Approach comes from beneath. A shark climbing gradually through the water column can enter the space below an animal without using the horizontal pursuit typical of faster predators. This geometry matters because prey must divide attention among many possible threats while swimming, feeding, or surfacing. The sleeper shark needs only to continue upward at a measured pace until the remaining distance becomes small. That pace is extraordinarily restrained, often less than half a mile per hour during ordinary movement. Low tail beat frequency and soft musculature reduce the amount of water displaced during approach. A rapidly accelerating predator pushes a pronounced pressure disturbance ahead of its body, information that prey, equipped with sensitive mechanoreceptors, may detect. The sleeper shark produces a weaker hydrodynamic announcement. Its enormous size advances without the turbulence expected from an animal carrying so much mass. Once proximity has done most of the work, feeding requires little pursuit. Opening the mouth rapidly expands the oral cavity and draws water inward, helping pull nearby prey toward the jaws. Contact can then be converted into a controlled cutting action. Observations of feeding sleeper sharks have documented characteristic rotations of the head, allowing the lower cutting teeth to work through tissue while the upper teeth maintain purchase. The animal does not need prolonged thrashing to remove a mouthful. The same method also makes the distinction between predator and scavenger unusually flexible. A living fish, an injured marine mammal, or a carcass settling through deep water can all enter the same sequence. Chemical traces lead the shark toward food, gradual movement closes the distance, and the jaws complete the encounter. Nothing about the approach requires the target to flee, remain still, or even be alive. A rare view of this behavior arrived off Hainan Island in 2025. Researchers placed a cow carcass at a depth of roughly 5,345 feet in the South China Sea and recorded Pacific sleeper sharks visiting it. The animals did not form the turbulent feeding frenzy familiar from surface imagery. Individuals approached, fed, withdrew, and returned with measured movements. Immediately before contact, researchers observed the sharks retracting their eyes, drawing the vulnerable organs inward as the head pressed close to food. Here, the apparent disadvantages of the species become parts of the same mechanism. Limited vision does not interrupt an approach governed by other information, and low speed reduces the warning carried through water. The shark does not win by matching the acceleration of its prey. It changes the encounter itself, replacing pursuit with positioning until speed has almost nothing left to decide. The most revealing record of a Pacific sleeper shark's hunting life is often found after the animal itself is gone. In the Gulf of Alaska, researchers examined the stomachs of 11 juvenile sleeper sharks and discovered a menu far broader than their unhurried appearance suggested. Giant Pacific octopus occurred in roughly 72% of the stomachs, while arrowtooth flounder appeared in about 63%. Unidentified bony fishes occurred in 45%, and squid in 36%. Pollock, rockfish, salmon, shrimp, hermit crabs, marine snails, and other flatfish added still more variety. Among those remains, octopus stood out. Giant Pacific octopuses are muscular, responsive animals capable of rapid jet-propelled escape, flexible enough to enter narrow crevices, and strong enough to resist many attackers. They also spend considerable time along the seafloor and rocky habitat, placing them within the sleeper shark's feeding range. Once approached closely, their soft bodies are particularly vulnerable to suction and a gripping bite. Their frequency in juvenile stomachs suggests that this is not an occasional encounter, but an important source of food during earlier life stages. Fish expand the picture beyond the bottom. Adult Pacific salmon have also appeared in sleeper shark stomachs. These are powerful swimmers adapted to sustained movement, schooling behavior, and rapid responses to nearby disturbance. Their presence matters because a healthy adult salmon is not the sort of prey usually associated with a sluggish predator. Yet the digestive evidence places the two animals in the same feeding event. Marine mammals make the contrast even greater. Research on harbor seals has provided evidence consistent with direct predation by Pacific sleeper sharks rather than simple consumption of carcasses. Harbor seals can reach roughly 25 miles per hour during short bursts and possess excellent underwater maneuverability. Speed alone should give them an overwhelming advantage during a conventional chase. The sleeper shark does not depend on a conventional chase. Larger sharks have yielded remains from stellar sea lions, animals that can reach well beyond 2,000 pounds in the largest males. Such material must be interpreted carefully because stomach contents do not always reveal whether prey was killed or scavenged. A sea lion found inside a shark establishes consumption, while circumstances surrounding its death may remain uncertain. Even so, the ability to process such a large marine mammal reveals the scale of food available to bigger sleeper sharks. Squid remains carry similar ambiguity. Beaks survive digestion long after softer tissues disappear, making them useful evidence of feeding but poor witnesses to the exact encounter. Large squid may be captured alive, consumed while injured, or scavenged after death. For an opportunistic predator, those categories need not be exclusive. A meal does not become less valuable because another animal killed it first. Taken together, the stomach records describe a predator without a narrow specialty. Small crustaceans, bottom-dwelling invertebrates, schooling fish, cephalopods, and marine mammals all enter the same dietary spectrum. The balance also appears to shift with body size. Juveniles rely heavily on octopus and fishes, while larger individuals gain access to increasingly substantial prey. That progression leaves the largest adults as an unresolved part of the food web. Their stomachs have never provided researchers with the same physical record available from younger animals. What can be established is already unusually broad. From hermit crabs moving across the bottom to seals crossing open water, the Pacific sleeper shark feeds across categories that rarely belong to a single hunting style. Its diet belongs not to a predator constrained by slowness, but to a generalist able to turn many forms of opportunity into food. Inside the body of a Pacific sleeper shark lies an organ whose scale changes the arithmetic of survival. The liver can account for as much as 30% of total body mass. In an exceptionally large shark weighing 2,000 pounds, that proportion could mean roughly 600 pounds of liver. An average adult human weighs around 170 pounds, so the organ alone could outweigh 3 people. Yet its importance is measured less by size than by what is stored within it. In this animal, anatomy becomes a reserve account, with buoyancy and stored energy concentrated in the same immense structure. Much of that mass consists of lipid-rich tissue, including oils known as alkyldiacylglycerols. These compounds distinguish sleeper sharks from many other deep-dwelling sharks whose livers may contain large amounts of squalene. Squalene has long attracted commercial interest and has been used in products including cosmetics. Alkyldiacylglycerols have not carried comparable economic value. That biochemical difference has helped make Pacific sleeper sharks less attractive as direct targets for liver oil fisheries, although they can still encounter fishing gear intended for other species. For the shark itself, those oils solve a basic problem of life without a swim bladder. Shark tissues are generally denser than seawater, creating a natural tendency to sink. Lipids in the liver are less dense, adding buoyant lift and reducing the muscular work required to remain within a useful depth range. The liver does not make the animal perfectly weightless, but it lowers the energetic price of moving through deep water. For a predator whose success depends on conserving energy, even a small reduction in continuous effort matters across days, seasons, and years. The same reservoir also serves as stored chemical energy. Food in the deep ocean can arrive irregularly, and large prey may be separated by long intervals. Lipids pack substantial energy into relatively little biological mass, allowing a sleeper shark to draw upon internal reserves when feeding opportunities decline. Precisely how long a Pacific sleeper shark can fast has not been established, but its physiology is suited to enduring prolonged gaps between meals, far better than a predator dependent on frequent feeding. That reserve accompanies another demanding part of its life: repeated movement through the water column. Tagged sleeper sharks have been recorded changing depth again and again, sometimes traversing hundreds or thousands of feet. A large oil-rich liver reduces the energetic burden of these movements by contributing buoyancy while also carrying the fuel that powers metabolism. Pressure changes with depth affect the animal as a whole, yet the liver remains fundamentally a buoyancy organ and energy store rather than a mechanical elevator. Its contribution is subtler, reducing costs that accumulate with every ascent and descent. Cold water adds another layer to this economy. Lower body temperatures, slow swimming, and conservative movement are associated with a low rate of energy use. Among sleeper sharks, such metabolic restraint accompanies remarkably slow life histories and exceptional longevity. Energy that a faster predator continually spends on acceleration and pursuit can remain stored here, available for maintenance, growth, movement, and the long intervals between successful feeding events. Seen this way, the liver is not merely an oversized organ tucked behind the shark's ribs. It is part of the physical foundation that makes the animal's lifestyle possible. Buoyancy reduces the cost of remaining at depth. Stored lipids soften the consequences of uncertain meals. A restrained metabolism stretches those reserves across time. The Pacific sleeper shark survives through an economy of expenditure, carrying within its own body a vast reserve that allows patience to function as physiology. Each day leaves a vertical trail through the Pacific, not on the seafloor, but through the water itself. As daylight changes above, entire communities shift their depth. Small fish, squid, and other animals descend or rise according to light, predators, and feeding opportunity. Pacific sleeper sharks participate in this immense daily rearrangement. Their movements follow a pattern known as diel vertical migration, an ascent through the water column during the darker hours followed by a return toward deeper water during daylight. Acoustic tagging in Alaskan waters has revealed how extensive those movements can be. Tracked sleeper sharks repeatedly traveled through hundreds of feet of depth in a single daily cycle. Much of their recorded time was spent between roughly 500 and 1,500 feet. Yet regular excursions carried them above 330 feet. One tagged individual went much farther. On multiple occasions, its transmitter placed it within roughly 7 feet of the surface. An animal commonly associated with deep slopes and cold offshore water had crossed nearly the entire vertical boundary, separating its usual habitat from the open air. The timing gives those journeys their biological meaning. During daylight, tagged sharks generally occupied deeper water. Through the afternoon, they began climbing, continuing upward as night settled over the ocean. Their ascent tended to be slower than their descent. That asymmetry fits an animal feeding along the way. Rising gradually allows it to pass through layers occupied by prey, while the faster downward movement returns it efficiently toward deeper daytime habitat once conditions near the surface change. Many potential prey species are making journeys of their own. Fish and squid often rise after sunset as reduced illumination changes the balance between feeding and exposure. Marine mammals also use different depths while traveling, resting, and searching for food. Instead of pursuing scattered animals across a vast three-dimensional ocean, the sleeper shark can move toward layers where living biomass becomes temporarily concentrated. Its nightly climb therefore functions as a meeting point between predator behavior and the daily schedule of an entire ecosystem. Direction matters as much as timing. The shark approaches these layers from beneath. A seal near the surface occupies a world bounded by air above and deeper water below, while schooling fish may be oriented toward movement occurring around their own depth. A large predator, rising gradually from underneath, enters from an angle that may provide little warning. Its low swimming speed produces relatively modest water disturbance, allowing an enormous body to reduce the hydrodynamic signals that might otherwise announce its arrival. Such travel carries an energetic cost, but the sleeper shark is unusually equipped to make that cost manageable. Its lipid-rich liver contributes buoyancy while storing substantial energy, and its low metabolic demands keep routine movement comparatively economical. Hundreds of feet of climbing each night need not resemble an athletic sprint. Spread across hours, the ascent becomes a measured investment, one that can be repaid when a fish, squid, or marine mammal comes within feeding range. The visits to the uppermost water are especially revealing. Reaching within several feet of the surface places a sleeper shark among animals that rest, travel, and feed there after dark. These records show that depth is not a fixed address for the species. Deep water provides daytime habitat, while the upper ocean can become part of its nocturnal feeding territory. Across each cycle, the shark traces the changing distribution of life rather than remaining attached to a single depth. Daylight draws it downward. Darkness permits a gradual rise. Prey gathers higher in the water, and the shark follows that living layer until its journey can end almost at the surface. Then the pattern reverses, carrying the animal back into deeper water as another daily migration closes. Reproduction leaves evidence even when the event itself remains hidden. Somewhere across the Pacific, mature sleeper sharks mate, embryos develop, and young enter the ocean. Yet, the reproductive lives connecting those stages have almost never been witnessed. Despite decades of fisheries encounters, research surveys, tagging programs, and deepwater observations, no confirmed pregnant Pacific sleeper shark has provided scientists with the embryos needed to establish gestation, litter size, or developmental timing. For an animal capable of becoming one of the largest predatory fishes in the sea, the beginning of life remains among its least accessible chapters. Researchers generally infer that Pacific sleeper sharks reproduce through aplacental viviparity, a mode found among several related sharks. Fertilization occurs internally, and embryos develop within the mother without a placental connection, drawing nourishment initially from yolk. The young are eventually born alive. Very small Pacific sleeper sharks indicate that newborns are around 16 inches long, a remarkably modest beginning for an animal that may eventually extend many feet through the water. Those young confirm that successful births are occurring, but almost everything surrounding them remains unresolved. Scientists do not know the normal litter size. Gestation length has not been established. A consistent birth season has not been identified. Even the location of pupping remains uncertain, although research in the Bering Sea has found aggregations of young sharks, and genetic evidence linking siblings has strengthened the possibility that parts of the region provide important nursery habitat. The missing adults are central to the problem. Fisheries surveys encounter Pacific sleeper sharks only imperfectly, and the largest mature animals appear to occupy habitats that ordinary sampling gear rarely reaches effectively. Deep continental slopes, trenches, and offshore basins create an immense three-dimensional refuge where nets and longlines sample only tiny fragments of available habitat. A shark can therefore exist within the range of modern instruments while remaining almost absent from physical collections. Cameras may record an animal that cannot practically be brought aboard, examined internally, and released or preserved for detailed reproductive study. Recent observations have added another intriguing pattern without resolving it. In 2025, cameras in the northern South China Sea recorded 8 Pacific sleeper sharks feeding at great depth, and every individual observed was female. Such segregation by sex occurs in many shark species, so the gathering cannot by itself identify a breeding or nursery area. It does show that females may concentrate in particular deep water habitats, far beyond the distribution scientists once considered typical, widening the geographic space in which reproduction might occur. A rare female from the Aleutian Islands illustrates how slowly the evidence accumulates. The shark measured about 14 feet long and reached researchers through what they described as sheer awesome luck. Another clearly immature female, studied through radiocarbon analysis, was already at least 35 years old, demonstrating how much time can pass before reproductive maturity. Researchers, examining the larger Aleutian specimen, preserved her enormous eye lenses, described as being the size of cantaloupes, because chemical records within those lenses may help reveal age, yet the reproductive information most desired remained unavailable. She was not carrying a litter that could reveal how many pups develop together or how far development proceeds before birth. This absence matters far beyond anatomical curiosity. Population models depend on the rate at which animals replace themselves. A species that matures late, produces few young, and reproduces infrequently can decline under fishing pressure far faster than abundance alone might suggest. For Pacific sleeper sharks, the essential values needed for those calculations remain uncertain. Births continue somewhere across an enormous ocean, but science still lacks the reproductive timetable that would reveal how slowly or how successfully this ancient predator renews its population. A shark's eye can preserve a date that the rest of its body cannot reveal. Deep within the lens are proteins formed during embryonic development. Once created, these proteins remain in place for life, retaining a chemical record of the carbon present when the animal first began growing. Scientists can measure carbon isotopes in the lens core and estimate when that tissue formed, turning an eye into a biological timestamp. The same radiocarbon approach helped establish the extraordinary longevity of the Greenland shark, whose oldest estimated individuals may approach 4 centuries. Applied to Pacific sleeper sharks, the method has begun exposing another unusually slow clock. A pilot investigation of a shark roughly 10 feet long indicated growth around twice as fast as that of its Greenland relative. Twice the pace sounds substantial until the comparison is understood. Greenland sharks grow with exceptional slowness, so doubling that rate still leaves the Pacific sleeper shark developing far more gradually than most large vertebrates. A female recovered from the Aleutian region gave researchers an especially revealing glimpse. She measured about 14 feet long, already the length of a substantial predatory shark, yet age work indicated that she was at least 35 years old and still reproductively immature. Across much of the animal kingdom, 35 years would place an individual well beyond the beginning of reproductive life. In this species, those decades may represent only an extended approach to adulthood. Researchers with the National Oceanic and Atmospheric Administration have suggested that sexual maturity may require 50 years or more. If that estimate is correct, a 35-year-old female could still have another 15 years before producing her first offspring. A sleeper shark born when one human generation was young might not reproduce until that generation had raised children of its own. Biological time stretches differently inside an animal whose growth is measured across decades. Its maximum lifespan remains unknown. No Pacific sleeper shark has been aged well enough to establish a confirmed upper boundary, and direct comparison with the Greenland shark cannot provide one. Still, the relationship is suggestive. The Greenland species grows at roughly half the estimated rate and can survive for centuries. Pacific sleeper sharks may therefore possess lifespans measured not merely in decades, but potentially in centuries as well. Federal researchers describe the species as exhibiting extreme longevity, while acknowledging that the oldest ages remain unresolved. That possibility changes the historical scale of an adult shark moving through the Pacific today. An individual reaching maturity now may have begun life in the 1970s or earlier. Older animals could have entered the ocean in the early 20th century, perhaps before commercial aviation became an ordinary part of human travel. Some may already have been decades old when engineers began building submersibles capable of entering the depths where sleeper sharks could finally be observed. Longevity, however, does not automatically protect a species. It can create a particular vulnerability when paired with late maturity. An animal requiring at least half a century before first reproduction cannot rapidly replace adults removed from the population. Its litter size remains uncertain, its reproductive interval is unknown, and every indication points toward a life history operating at a slow tempo. Population loss under those conditions accumulates across generations that humans may barely notice. A mature shark removed today represents decades of growth that cannot be replaced next season, or even within several decades. The mathematics of survival, therefore, runs on the same extended clock as the animal itself, with each adult carrying many years of biological investment through the deep Pacific. There is a boundary in the Pacific where ordinary fisheries science effectively stops. Longlines, trawl nets, and survey equipment can work at impressive depths, but in most Pacific fishing contexts, their practical reach extends to roughly 6,600 feet. Mature Pacific sleeper sharks are believed to spend much of their lives far below that boundary, between approximately 9,800 and 19,600 feet. Even at the shallow end of that suspected adult range, thousands of feet of ocean separate the sharks from the tools normally used to study them. That separation shapes nearly everything scientists think they know about the species. Population models depend on observations, captures, measurements, and repeated sampling. For Pacific sleeper sharks, those foundations are drawn overwhelmingly from younger animals occupying accessible water. Mature adults, the reproductive core that ultimately determines whether the population grows or declines, have never been sampled systematically across their range. The National Oceanic and Atmospheric Administration has acknowledged that a large portion of the population remains unseen and unsampled. That limitation reaches beyond uncertainty about abundance. Without representative adults, researchers cannot reliably determine age structure, reproductive output, natural mortality, or the proportion of mature animals within the population. The missing sharks are precisely the individuals carrying the information most important for understanding long-term survival. Nearly every specimen available for detailed examination has entered science accidentally. Juveniles and subadults become entangled or hooked in fisheries, pursuing sablefish, pollock, Pacific cod, and other commercially valuable species. Researchers can measure those animals, collect tissue, inspect stomach contents, study parasites, and examine growth. Yet this creates a powerful sampling bias. The most accessible members of the species are also its youngest and least representative of the mature population hidden below. Direct observation has offered only scattered openings into that deeper world. Large sleeper sharks have appeared during submersible and camera deployments in places such as Suruga Bay, waters around Hawaii, and, more recently, the South China Sea in 2025. These encounters are valuable precisely because they are uncommon. They are not the result of a broad, repeated census. A camera enters suitable habitat for another purpose, and sometimes a sleeper shark passes through its field of view. Systematic work at such depths demands specialized vehicles, pressure-resistant cameras, deep-rated landers, ships, crews, and long periods of expensive sea time. Searching thousands of square miles for a widely dispersed predator becomes an undertaking very different from conventional fisheries research. Historically, Pacific sleeper sharks have also received less research attention than commercially important species, leaving technology, cost, and scientific priority layered together as barriers. Genetics has made this absence of information more consequential. Recent analyses indicate that Pacific sleeper sharks may form a single broadly distributed genetic stock across the Pacific rather than several isolated regional populations. If that interpretation holds, animals separated by enormous distances belong to one connected biological population. Losses occurring in one region cannot simply be assumed to affect an independent local group while distant populations remain untouched. There may be no separate reservoir capable of insulating the species from pressure elsewhere. So conservation proceeds with an unusual imbalance. Science possesses fragments from younger sharks, occasional images from great depth, genetic clues spanning an ocean, and incomplete records from fisheries. Beneath all of them remains the population that matters most, mature animals living beyond routine sampling, reproducing by mechanisms still largely unknown. The Pacific sleeper shark is difficult to understand, not simply because encounters are uncommon, but because most of its adult existence unfolds beneath the practical reach of the science attempting to count it. A cow carcass resting 5,345 feet beneath the South China Sea was meant to imitate something much larger. Researchers working off Hainan Island in 2025 were studying whale falls, the temporary deepwater ecosystems created when a large body sinks and transfers its stored organic matter to the seafloor. The experiment was designed around decomposition, scavengers, and the gradual redistribution of nutrients. Pacific sleeper sharks were not part of the plan. Until those cameras began recording, the species had never been documented there. Then large gray bodies entered the frame. Multiple Pacific sleeper sharks had found the carcass, thousands of miles south of the range where scientists had expected them. Their presence carried a geographical surprise greater than a simple new dot on a distribution map. Sleeper sharks were strongly associated with colder northern waters, yet here they were in a tropical region, using the deep South China Sea as viable habitat. Temperature at the surface mattered less than the cold conditions preserved far beneath it. The footage also captured behavior that specimens hauled from fishing gear could never reveal. Several sharks approached the food and fed without the violent competition commonly associated with gatherings of large predators. Individuals moved in, took their place at the carcass, fed, and withdrew. There was no need for explosive acceleration. Even around concentrated food, their movements retained the measured economy that defines the species elsewhere. At the instant of feeding contact, another detail became visible. The sharks retracted their eyes into their heads. This protective movement shields vulnerable tissue when the face presses against food and other animals, and it offered a rare view of how little visual information matters during the final act of feeding. Contact was guided by senses already carrying the animal accurately to its target. The eyes could be physically withdrawn at precisely the moment a surface predator might depend upon them most. Every sleeper shark recorded at the site was female. A single observation cannot establish the purpose of an entire region, and the finding does not prove that the South China Sea is a breeding ground. Yet the pattern is scientifically useful. Sex-based differences in habitat use occur among many sharks, and an all-female gathering raises the possibility that certain deep regions function as feeding areas, reproductive habitat, or seasonal destinations used disproportionately by females. Geography may be the larger lesson. The South China Sea is semi-enclosed and tropical at the surface, an unlikely setting according to the older picture of Pacific sleeper shark distribution. Finding several individuals there suggests that suitable deep habitat can connect regions that appear very different when judged from above. Ocean basins contain layers of temperature, pressure, prey, and currents that do not follow the ecological boundaries drawn from surface conditions. That possibility becomes more significant beside genetic evidence indicating that Pacific sleeper sharks may belong to one broadly connected stock across the Pacific. Rather than isolated regional groups remaining near familiar northern habitats, individuals may move through a far larger network of deep-water corridors. The sharks off Hainan Island therefore represent more than a range extension. They reveal how incomplete the visible map may be for an animal whose movements occur mostly beyond routine observation. The species was formally described in 1944. More than 80 years later, cameras documented it living in a sea where no confirmed record had placed it before. During all those intervening decades, Pacific sleeper sharks may have been crossing these waters beyond the reach of ordinary surveys, following food through deep layers that humans rarely observe. The discovery did not show an animal entering a new world. It showed science, finally noticing part of a world the shark may have known all along. Pressure leaves a chemical signature inside the Pacific sleeper shark. In the cold depths where this animal spends much of its life, its tissues contain unusually high concentrations of trimethylamine oxide, a compound shared with its Greenland shark relatives and many other deep-water fishes. Its purpose is fundamental. Extreme pressure can distort proteins and interfere with the molecular machinery that keeps cells functioning. Trimethylamine oxide helps stabilize those proteins, allowing biological processes to continue under conditions far removed from the surface. The same chemistry becomes troublesome after death. As tissues begin to break down, bacterial activity can convert trimethylamine oxide into trimethylamine and related compounds. Fresh sleeper shark flesh can therefore become unsafe to consume without careful processing. Accounts involving sleeper sharks describe effects resembling severe intoxication, including disorientation, impaired coordination, and other neurological disturbances. Dogs given sleeper shark meat have also been reported developing neurological symptoms, reinforcing a danger long recognized by northern coastal communities that encountered these animals. This does not mean the shark carries venom or deliberately manufactures a poison for defense. The chemistry exists because of the physical demands of deep water. Toxicity to a consumer is an indirect consequence of an adaptation serving another purpose entirely. Inside the living shark, trimethylamine oxide supports cellular stability. Once the animal dies and its tissues change, that useful compound becomes part of a very different chemical process. Its Greenland shark relative provides the best-known comparison. Greenland shark flesh has traditionally been rendered edible in Iceland through prolonged processing and fermentation, producing a food called hakkerl. Preparation takes months and allows undesirable compounds to dissipate or transform before consumption. No comparable large commercial tradition developed around Pacific sleeper shark meat. Processing an animal with little established market demand simply offered few economic rewards. The liver provides no strong incentive either. Many sharks have historically been exploited for oils rich in squalene, a substance once widely valuable in products ranging from industrial lubricants to cosmetics. Pacific sleeper sharks instead store much of their liver oil as alkyldia sialglycerols. These lipids are useful to the shark, but they have never carried comparable commercial importance. Its fins likewise have not made the species a major target of dedicated fisheries. Flesh requiring substantial treatment, liver oil with limited value, and relatively unimportant fins combine into an animal that fishers generally have little reason to pursue deliberately. That economic obscurity has offered a peculiar kind of protection. Direct fishing pressure remained lower than it might have been if the shark possessed valuable meat or oil. Yet the same lack of value also helped keep it outside the center of fisheries research. Commercially important species generate landing records, biological sampling, stock assessments, dedicated surveys, and sustained funding because governments and industries need to know how many can be harvested. An animal commonly discarded as unwanted bycatch creates a thinner trail of information. For years, that thin record shaped conservation knowledge. The International Union for Conservation of Nature previously treated the Pacific sleeper shark as data deficient, reflecting how little evidence existed for judging population status. Economic indifference was not the only reason for that uncertainty, since extreme depth and elusive adults remain formidable obstacles, but it helped determine which questions received money, vessels, personnel, and years of attention. The chemistry that steadies this shark beneath immense pressure therefore carries an unexpected human consequence. It reduces the usefulness of its flesh, contributes to its low commercial appeal, and helped leave generations of Pacific sleeper sharks largely outside sustained scientific attention. An adaptation that makes life possible in deep water also helped make the animal economically easy to overlook. A predator weighing hundreds or even thousands of pounds can still become prey. For the Pacific sleeper shark, that reversal appears uncommon, but it matters because the few animals capable of attacking it help define where this shark truly stands in the North Pacific food web. Size, deep-water chemistry, and a formidable feeding apparatus narrow the field considerably. They do not remove it. Killer whales provide the clearest biological example. In North Pacific waters, sharks are documented components of the diets of some killer whale populations, and sleeper sharks have been identified among their prey. Certain killer whales also develop pronounced tooth wear associated with feeding on sharks, whose abrasive skin can gradually grind down enamel. A Pacific sleeper shark presents a large target, yet killer whales possess advantages few marine predators can combine: speed, coordinated group behavior, powerful jaws, and the ability to learn specialized hunting techniques. Other large sharks may represent a narrower threat, particularly to younger sleeper sharks. A very large great white shark, could plausibly overpower a juvenile encountered within overlapping habitat, as could another sufficiently large predatory shark. Specific predation on Pacific sleeper sharks by great whites has not been confirmed, however, so this remains biological possibility rather than documented behavior. For mature sleeper sharks, the list of plausible attackers becomes shorter as both body size and habitat change. Depth itself becomes the adult shark's strongest refuge. Once a sleeper shark moves below roughly 6,600 feet, the familiar predators of upper ocean ecosystems can no longer reliably follow. Killer whales cannot dive remotely close to the deepest habitat attributed to mature adults. Great white sharks also remain far above those abyssal zones. At depths approaching 20,000 feet, separation from surface predators is created not by speed or defensive weaponry, but by an immense vertical barrier of water. Younger sharks do not yet possess that same protection. Juveniles occur much higher in the water column, sometimes between roughly 160 and 1,000 feet, placing them within reach of marine mammals and large predatory fishes. This creates a long, vulnerable interval in a life history already measured at an unusually slow pace. If sexual maturity requires 50 years or more, every shark lost before reproducing represents decades of growth that cannot be rapidly replaced by another generation. The most consistently recorded source of mortality, however, does not arrive with teeth. Commercial fishing gear intersects precisely with the depths occupied by younger Pacific sleeper sharks. Longlines and other fisheries targeting sablefish, pollock, Pacific cod, and North Pacific groundfish capture sleeper sharks incidentally. Because the species has little commercial value, these animals are generally unwanted and are commonly discarded rather than retained. Release does not necessarily erase the encounter. A deepwater shark brought upward on fishing gear experiences, handling, exhaustion, injury, and a rapid environmental change before returning to the sea. Reliable post-release survival estimates for Pacific sleeper sharks remain limited, making the true mortality associated with bycatch difficult to calculate. An animal disappearing beneath the surface after release may survive, or its injuries may become consequential later, beyond observation. Ecologically, the Pacific sleeper shark, therefore, occupies a position near the top rather than beyond predation altogether. Few natural hunters can challenge a large adult, and extreme depth eventually removes most of them from its world. Yet juveniles spend decades within a more crowded field of risk. Among those risks, fishing gear is uniquely persistent. Evolution equipped the sleeper shark for pressure, cold, scarcity, and biological predators. It never had reason to evolve around hooks descending from ships above. For decades, one official phrase followed the Pacific sleeper shark through conservation records: data deficient. It did not mean the species was secure. It meant science lacked enough information to judge its condition with confidence. Then, in 2021, the International Union for Conservation of Nature moved the shark into near-threatened status. The change came not from a sudden abundance of knowledge, but from evidence accumulating around something harder to ignore. Parts of the population appeared to be declining. Fisheries records offered some of the clearest traces. In Taiwan, a small deepwater longline fishery had once encountered Pacific sleeper sharks with some regularity. By 2019, those encounters had become rare. Particularly concerning was the disappearance of larger subadults and adults, the animals nearest to or already participating in reproduction. Elsewhere, across portions of the North Pacific, survey catch rates also showed measurable declines. None of these records could reveal the entire population, but together they suggested that absence was becoming part of the data. For a species with such slow life history, even modest losses carry unusual weight. National Oceanic and Atmospheric Administration researchers have compared its vulnerability with that of the spiny dogfish, a relative capable of living around 100 years. That species can tolerate harvest rates of only about 3%. Pacific sleeper sharks may mature later, live longer, and replace themselves even more slowly, implying an allowable rate of loss that could be smaller still. Yet, the actual bycatch mortality remains uncertain, and so does the level the population could sustain. This uncertainty sits at the center of management in United States waters. Pacific sleeper sharks are incorporated into federal stock assessments and fishery management systems, but the calculations depend heavily on animals encountered where fishing gear can reach. Those encounters are dominated by younger sharks. Mature adults remain largely outside systematic sampling, leaving the reproductive core of the population poorly represented in the models intended to protect it. Researchers have summarized the problem in plain terms. Effective management depends upon understanding an animal that remains exceptionally difficult to observe. Time makes that uncertainty more consequential. A fish population that matures rapidly can sometimes rebuild after heavy losses within years. The Pacific sleeper shark works on another calendar. If first reproduction comes only after roughly 50 years or more, removing a mature animal erases an investment extending across half a century before its reproductive contribution can even begin. Should abundance fall far enough to require deliberate rebuilding, recovery could occupy many decades and potentially much longer, depending on litter size, reproductive frequency, and survival of young, all parameters still inadequately known. There is no directed industrial hunt responsible for most of this pressure. In Alaska, fisheries bycatch accounts for most documented Pacific sleeper shark mortality. Gear deployed for commercially valuable groundfish can catch sleeper sharks simply because both occupy portions of the same water column. The shark may have no market waiting for it and no vessel searching specifically for it, yet it can still be removed from a population whose replacement rate is extraordinarily slow. That gives the conservation problem an unusual shape. The Pacific sleeper shark survived immense changes in ocean temperature, shifting coastlines, glacial cycles, and ecological rearrangements over evolutionary time. Its present pressure does not require anyone to pursue it intentionally. Economic activity aimed at other animals is enough. Hooks descend into habitat used by young sleeper sharks, and each accidental capture enters a population equation whose most important numbers remain unknown. By the time those numbers become clear, the decades required to replace what was lost may already have begun. The name came from a misunderstanding made at the surface. Naturalists encountering members of the sleeper shark family saw animals that appeared sluggish, heavy, and strangely subdued. The family name Somniosidae traces back to the Latin word somnus, meaning sleep, and the impression became permanent. Yet many of those sharks had just been hauled upward from deep water, subjected to rapid changes in pressure, temperature, and surroundings. What looked like natural lethargy may instead have been the behavior of an animal physiologically displaced from the world in which its body was built to function. That first impression shaped more than vocabulary. For generations, sleeper sharks were widely portrayed as scavengers, animals drifting across the seafloor and accepting whatever carrion happened to appear. Their low swimming speeds seemed incompatible with active predation, so researchers often interpreted their feeding ecology through that assumption. A slow shark, in this view, could consume large animals only after something else had killed them. The label helped turn an observation into an expectation, and the expectation persisted even as evidence accumulated against it. That assumption also helped keep the family at the margins of research, where animals considered passive scavengers attracted less attention than more visibly active hunters. Stomach contents eventually made the old picture difficult to maintain. Pacific sleeper sharks have contained harbor seals, stellar sea lions, adult salmon, squid, octopus, and other prey whose lives are defined by movement. Some remains can certainly come from scavenging, particularly when large carcasses become available, but scavenging alone cannot comfortably explain the entire diet. Harbor seals are alert swimmers. Adult salmon move powerfully through open water. Living cephalopods react rapidly to nearby disturbance. A predator repeatedly consuming such animals is doing more than waiting for death to deliver food. Its slowness becomes more coherent when viewed through temperature rather than temperament. Much of the Pacific sleeper shark's habitat lies near 36 degrees Fahrenheit, where an ectothermic animal receives little external heat to accelerate its metabolism. Chemical processes within muscle and other tissues proceed slowly. Sustained speed therefore carries a substantial energetic price. For a predator living where meals may be widely separated, spending large reserves simply to move faster would often return little benefit. Efficiency offers another solution. A sleeper shark can conserve energy during approach, exploit sensory information that does not depend on vision, close the final distance gradually, and commit its greatest effort only when prey is within striking range. The strategy does not require winning a prolonged chase. It requires arriving close enough without provoking an escape soon enough to matter. In cold, deep water, patience becomes part of the mechanics of predation. Natural selection has no preference for speed by itself. It favors whatever combination of traits leaves descendants. Across geological time, the North Pacific has held predators built for acceleration, endurance, pursuit, and powerful bursts of movement. Lineages have appeared and vanished as oceans changed around them. Sleeper sharks persisted with another arrangement, conserving energy, tolerating scarcity, exploiting opportunities across an unusually broad menu, and moving at a pace matched to the physical conditions surrounding them. Seen this way, the word sleeper reveals more about human observation than shark behavior. People watched an animal after removing it from cold, pressurized depths and named it for the weakened motion they could see. Down where that name has no meaning, slowness is not passivity. It is an economical way to cross a landscape where every calorie matters, an approach refined long before humans began describing the ocean. The Pacific sleeper shark does not move slowly because it has failed to become fast. It moves slowly because in its world, speed is often an unnecessary expense. Long before familiar coastlines took their present shape, the ancestry of sleeper sharks was already being written into the history of the oceans. Fossil evidence places somniocid sharks deep within geological time, with the broader lineage extending back toward the Cretaceous world. That era ended about 66 million years ago, in a mass extinction that erased the non-avian dinosaurs and transformed marine ecosystems across the planet. Ocean food webs fractured, dominant groups vanished, and surviving lineages entered a radically altered world. Sleeper sharks endured beyond that boundary, carrying a deep-water way of life into the oceans that followed. Their persistence continued through repeated changes in climate that remade the surface of Earth. Around 20,000 years ago, during the last glacial maximum, Enormous volumes of water were locked in continental ice. Sea level stood roughly 400 feet lower than today, exposing broad continental shelves and redrawing coastlines around the Pacific. Habitats near shore shifted or disappeared as currents, temperatures, and productive zones moved. Far below those changing margins, cold water remained a familiar realm. For animals adapted to low temperatures and great pressure, glacial conditions did not create the same upheaval experienced by many communities closer to sunlight. Much earlier, the planet had moved in the opposite direction. Around 56 million years ago, during the Paleocene-Eocene thermal maximum, global temperatures rose sharply over a geologically brief interval. The warming reached into the oceans, altering circulation, chemistry, oxygen conditions, and the distribution of marine life. Deepwater ecosystems were not insulated from the disturbance. Extinctions occurred among organisms living on and within the seafloor, while surviving species shifted their ranges as suitable conditions moved. Yet the sleeper shark lineage persisted through this warmer chapter as well. Such endurance gives its modern biology a different scale. Energy conservation, tolerance of cold water, broad feeding habits, sensitivity to non-visual signals, and movement across large depth ranges are not merely useful traits in the present Pacific. They belong to an evolutionary pattern shaped through oceans that repeatedly changed around the animals inhabiting them. Specialists can flourish when conditions favor a narrow way of life, but specialization can become a trap when prey disappears or habitat shifts. A generalist able to move through depth, conserve fuel, and accept many kinds of food carries options into periods of instability. One comparison makes that persistence especially vivid. Otodus megalodon, the immense predatory shark that once occupied warm and temperate seas, disappeared around 3.6 million years ago. Its extinction followed major ecological changes involving climate, prey communities, and competition. Ancestors and relatives of modern sleeper sharks were already present while Megalodon hunted marine mammals across ancient oceans. When that enormous predator vanished, the deepwater lineage continued, without requiring the speed, enormous energetic demand, or specialized ecological position of the more famous giant. Across these spans of time, the deep ocean offered something rare on a changing planet: relative environmental continuity. Temperatures and currents shifted, oxygen levels changed, and even the deep sea experienced major disruptions. But its cold, pressurized character remained more stable than the sunlit margins above. Sleeper sharks evolved within that slower physical world, matching their metabolism and behavior to conditions that often changed less abruptly than coastal ecosystems. Their survival, then, is not evidence of an animal untouched by history. It is evidence of a lineage repeatedly passing through history because its biology allowed room for change. Ice advanced and retreated, seas warmed and cooled, predators rose, dominated, and disappeared. The sleeper shark lineage remained in the deeper Pacific, conserving energy, finding food, and reproducing on timescales almost detached from human experience. For millions of years, that strategy has carried it through transformations that erased countless other forms of marine life. Its greatest strength has always been endurance, built for an ocean where survival belongs not necessarily to the fastest hunter, but to the one that can continue. The predators most familiar to us arrive with motion. A great white shark can surge at roughly 25 miles per hour. A sailfish can approach 68. On land, a cheetah can reach about 75. These animals helped shape a familiar picture of predation, one in which success belongs to the hunter capable of closing distance fastest. Far below that world, the Pacific sleeper shark moves at less than half a mile per hour and still takes harbor seals. Its existence separates speed from the idea of predatory competence. Speed works beautifully where prey can be seen across open space and pursuit determines the outcome. It is one solution shaped by particular conditions, not a requirement imposed on every hunter. In deep water, acceleration carries an energetic cost, while visibility offers little reward. The sleeper shark approaches the problem differently. Instead of paying for prolonged pursuit, it minimizes expenditure until the final encounter. A seal may be vastly faster in absolute terms, yet speed matters only after danger has been detected with enough time and distance to escape. Our second assumption comes from our own anatomy. Human beings organize much of their understanding through sight, so hunting without useful vision can seem like hunting without information. The deep Pacific reverses that hierarchy. There, electroreception can reveal the weak electrical activity produced by living muscle and nerves. The lateral line registers minute changes in water movement and pressure. Neither depends upon reflected light. A prey animal can blend visually into its surroundings, but it cannot stop its heart, muscles, and movement from altering the physical field around it. For the sleeper shark, these senses are not reduced replacements for eyesight. They fit the habitat more precisely than vision could. Darkness cannot conceal an electrical signal from a receptor designed to detect it. Camouflage cannot erase a pressure disturbance traveling through water. The very conditions that remove visual information leave other channels available, and evolution has equipped the shark to read them. Size creates another misleading expectation. A huge predator appears as though it should require a constant stream of large meals. That logic fits animals whose bodies burn energy rapidly. The sleeper shark carries a different budget. Cold surroundings suppress metabolic demand. Movement remains economical, and enormous lipid reserves store energy for lean intervals. A body weighing thousands of pounds therefore does not automatically imply the feeding requirements of a comparably sized warm-blooded predator. Sparse prey can support a large hunter when the cost of remaining alive is exceptionally low. Even rarity becomes difficult to judge through human experience. Pacific sleeper sharks remain among the least understood large predators on Earth, yet obscurity does not demonstrate scarcity. Their known distribution spans an immense portion of the North Pacific, with newer observations extending expectations farther across the Pacific Basin. Much of their habitat lies beyond ordinary observation and sampling. An animal can be widespread while remaining almost absent from cameras, nets, laboratories, and human memory. That distinction matters because conservation begins with knowing what ignorance means. Few observations can indicate a small population, but they can also indicate that researchers are looking through a narrow window into an enormous, inaccessible habitat. Confusing those possibilities can distort both risk assessment and ecological understanding. The Pacific sleeper shark, therefore, does not violate the rules of predation. It exposes how narrowly those rules were written. Humans learned first from hunters occupying bright, accessible environments, where speed, vision, and frequent feeding are easy to witness. Deeper water offers another set of physical conditions and another set of successful solutions. The sleeper shark belongs completely to that system, not as an exception, but as evidence that predation has always been broader than the part of it we could see. At 300 feet below the Pacific, sunlight has already begun surrendering its colors. Descend toward 1,000 feet, and the remaining blue fades toward the edge of usefulness. For a Pacific sleeper shark moving through deeper water, vision becomes only one faint channel among several, and eventually an almost irrelevant one. Its surroundings are not empty of information. They are crowded with signals that human senses were never built to notice. Pressure increases steadily with descent, reaching roughly 30 times surface pressure near 1,000 feet. Temperatures in much of the shark's deep habitat hover only a few degrees above the freezing point of fresh water. Yet neither condition represents an intrusion into the animal's life. Its tissues, chemistry, and metabolism function within them. The sensory world surrounding that body is carried through water as vibration, dissolved molecules, pressure disturbances, and weak electrical fields. Along the head are specialized receptors called ampullae of Lorenzini. Every living animal generates electrical activity as nerves, fire, and muscles contract. Living prey therefore produces information that does not depend on illumination. The signal is faint, but seawater conducts electricity, allowing specialized receptors to register nearby biological activity with precision. At close range, the sleeper shark can detect biological presence even when useful visual detail has disappeared. Another stream of information arrives through the lateral line, a sensory system running along the body and responding to water displacement. Movement leaves structure behind it. A tail stroke pushes water. A turning fish creates pressure changes. A diving mammal disturbs the surrounding flow. These traces can reveal direction and motion, allowing the shark to interpret nearby activity without needing a visible outline. Smell extends that awareness beyond immediate movement. Dissolved chemicals released by tissue, waste, blood, or decaying material travel with currents, forming irregular trails through the water. Following it requires repeated sampling as concentration shifts. For a predator adapted to conserving energy, this offers another way to locate food without racing through enormous volumes of water. Sound adds still another layer. Deep water remains acoustically connected across great distances. Cetacean calls, fish, crustaceans, ships, storms, and geological activity all contribute to that soundscape. The shark's inner ear detects vibration and low-frequency sound, while its lateral line is especially useful for nearby water movement. Together, these systems make darkness far less limiting than human intuition suggests. A harbor seal entering this realm carries its own sensory advantages, but the sleeper shark approaches with remarkably little expenditure. Slow movement reduces the disturbance produced ahead of its body and may make its arrival difficult to detect until the distance has narrowed. The same pace that appears ineffective from a surface perspective becomes useful when concealment depends partly on minimizing hydrodynamic disturbance. Cold water shapes the entire exchange. It generally contains more dissolved oxygen than warmer water, while low temperatures reduce metabolic demand in an ectothermic animal. Chemical cues can persist and disperse according to currents, and the dense aquatic medium carries mechanical disturbances efficiently. Together, these conditions favor sensitivity and energy conservation over speed. Humans describe these depths with words drawn from human vulnerability: crushing pressure, near freezing water, darkness, isolation. Those descriptions are accurate for a body designed for land and air, but they are not universal descriptions of experience. The Pacific sleeper shark inhabits the same conditions through another sensory architecture. Where human eyes lose the landscape, electrical fields, vibrations, dissolved chemicals, and pressure changes continue carrying information. For this shark, the deep Pacific is not a diminished version of the surface. It is a complete world, and its body is built to read it. There are species for which science possesses missing details, and there are species for which entire chapters of biology remain unwritten. The Pacific sleeper shark belongs to the latter. Even after decades of encounters across the Pacific, some of the most basic facts needed to describe its life cannot yet be stated with certainty. Its maximum confirmed lifespan is unknown. Its litter size is unknown. Gestation length has never been established. Mating behavior has never been observed, and the depth or habitat in which females give birth remains unidentified. Nursery areas for pups can only be inferred from where young sharks appear. The total population across the species range is unknown, as is its maximum depth. Records suggest habitat use extending toward roughly 20,000 feet, but the true boundary has not been confirmed. Movement carries similar uncertainty. Researchers do not yet know whether mature Pacific sleeper sharks follow regular migration corridors across the ocean basin, return seasonally to particular regions, or spend long periods within comparatively restricted, deep-water territories. Even diet becomes less certain with age. Much of the physical evidence from stomach contents comes from younger animals accessible to fishing gear, leaving the full feeding ecology of mature adults largely beyond direct examination. Whether these sharks produce sounds for communication is also unknown. The contrast becomes clearer beside predators that have received sustained scientific attention. Great white sharks can be tracked across oceans. Researchers have identified important nursery regions, documented movement patterns, studied reproductive biology, examined diets across life stages, and built increasingly detailed population models. Important uncertainties remain even for them, but there is enough information to connect behavior, reproduction, habitat, and conservation. For the Pacific sleeper shark, those connections are fragmentary. A predator that may attain extraordinary size is still managed using information drawn disproportionately from the younger animals that enter fisheries. The mature population occupies the part of its life history least accessible to direct study, precisely where many of the measurements needed for conservation must come from. Researchers with the National Oceanic and Atmospheric Administration have therefore emphasized several priorities. Adult age and growth must be resolved more precisely. Reproductive biology remains essential, along with the movement ecology of mature animals and the degree of population connectivity across the Pacific. Each missing measurement affects the others. Without reliable age data, growth models remain uncertain. Without reproductive rates, recovery potential cannot be calculated confidently. Without movement data, the geographic scale at which protection should operate remains difficult to define. The barrier is partly technological. Studying mature animals may require remotely operated vehicles capable of sustained work thousands of feet beneath the surface, where pressure challenges electronics, housings, cables, batteries, and instruments. Tagging presents another problem. A device must survive extreme depth, remain attached to a massive moving shark, record useful information, and eventually transmit or return those observations. Sampling must ideally gather biological material without hauling an animal through enormous pressure, changes merely to examine it. Pieces of that capability already exist. Deep ocean vehicles can reach the necessary depths, advanced tags continue improving, and environmental genetic techniques can detect species from traces left in seawater. What does not yet exist is a sustained, systematic research program applying those tools across the Pacific sleeper shark's immense range. So the imbalance remains. Science can describe parts of its anatomy, chemistry, prey, distribution, and extraordinary adaptations, yet the architecture of its life is still incomplete. The unknowns are not obscure details around the edges. They include birth, maturity, movement, abundance, reproduction, and death. Decades of limited economic interest and formidable depth have left one of the Pacific's largest predators present in scientific records, yet still only partly visible to science. The map suggests abundance before the records suggest absence. Across the North Pacific, the Pacific sleeper shark occupies an immense geographic space, extending from waters around Japan through the Bering Sea and Alaska, then south along the western coast of North America toward Baja California. Recent observations have placed it in the South China Sea, while historical records attributed to the species reach as far as Antarctic waters. Few large predators are associated with such an enormous sweep of ocean, yet direct encounters remain remarkably scarce. That contrast has shaped nearly everything science knows about the animal. Most specimens were never deliberately sought. A young shark takes bait intended for sablefish. Another enters a trawl pursuing pollock. Occasionally, a body reaches shore and provides tissue, measurements, or anatomy that researchers would otherwise struggle to obtain. Each encounter adds another fragment, but the circumstances are governed more by chance than by a planned meeting between scientist and shark. Purposeful observation is rarer still. Decades of submersible exploration have placed cameras into deep Pacific habitats for geology, ecology, fisheries, research, and the study of other animals. Pacific sleeper sharks have appeared only sporadically. A large individual was recorded in Suruga Bay in 1989. Other footage emerged from waters around Hawaii. In 2025, cameras in the South China Sea captured several more. Additional observations exist, but the total remains extraordinarily small compared with the scale of the shark's suspected distribution. Fishing records create their own distortion. Animals encountered within the reach of hooks and nets tend to be juveniles or subadults occupying shallower portions of the water column. They provide measurements of growth, diet, body condition, and distribution, but they represent only an early portion of a life that may continue for generations. Mature animals remain largely absent from samples. The population seen by fisheries is therefore not a miniature version of the whole population. It is a narrow age window made visible by where fishing equipment can operate. Tagging has begun to reveal movement, although the sample sizes show how early that work remains. A California study reported in 2024 followed only 4 Pacific sleeper sharks for limited periods. Their movements offered valuable evidence about behavior and habitat use, yet 4 individuals cannot describe an ocean-wide population. Every track is useful precisely because so few tracks exist. The species also gives researchers few predictable opportunities to improve those numbers. Pacific sleeper sharks are not known to gather in enormous schools or return to accessible surface locations where hundreds can be counted. They do not announce themselves through regular breaching, conspicuous feeding events, or dependable coastal aggregations. Much of their existence unfolds within a 3-dimensional expanse where an animal can pass miles from the nearest instrument and leave no visible trace behind. Presence, in this case, has an unusual meaning. A region may support sleeper sharks for years without producing a confirmed sighting. A baited hook may intersect one animal and create the impression of rarity, while others remain far below or beyond the fishing grounds. A camera can illuminate a tiny volume of water for hours and record nothing, even while suitable habitat extends for thousands of square miles around it. This is how a predator can occupy much of the largest ocean on Earth while remaining almost absent from human experience. Its record is assembled from accidental hooks, brief camera encounters, stranded bodies, and a handful of electronic tracks. The Pacific sleeper shark exists across the map like a word hidden within a vast page, present all along, but visible only when circumstance places our attention on exactly the right line. There is a particular kind of loss that leaves almost no visible absence. A population can thin beneath thousands of feet of water while ships continue crossing above it. Fisheries continue working, and scientific records continue accumulating. For the Pacific sleeper shark, that loss is emerging through fragments, fewer encounters, fewer large animals, and an increasingly troubling pattern in places where adults were once caught more regularly. Deep water longline records from Taiwan offer one such fragment. Pacific sleeper sharks that had appeared in catches became rare by 2019, with the reduction especially apparent among large subadults and adults. Elsewhere, across portions of the North Pacific, survey catch rates have also shown declines. These signals helped move the species from data deficient to near threatened on the International Union for Conservation of Nature Red List. The change reflected not complete knowledge, but enough evidence of deterioration to make continued uncertainty impossible to ignore. Commercial fishing creates another layer of uncertainty. Sleeper sharks encounter sablefish, longlines, pollock trawls, and fisheries associated with Pacific cod. Some are discarded alive after capture, yet release does not necessarily mean survival. A shark hauled rapidly upward from roughly 1,300 feet undergoes an enormous reduction in surrounding pressure while also enduring capture, stress, handling, temperature change, and possible injury. Unlike many bony fishes, sharks lack gas-filled swim bladders that cause classic barotrauma, but deep-dwelling species can still suffer severe physiological stress. For Pacific sleeper sharks, reliable post-release survival estimates remain scarce. That missing number matters because this species appears built around extremely slow replacement. An adult female removed from the population may embody 50 years or more of growth before reaching reproductive maturity. Whatever reproductive contribution might have followed across her remaining lifespan disappears with her. A rapidly reproducing fish can sometimes compensate for losses through abundant young. A sleeper shark cannot simply accelerate a life history shaped by cold water, slow growth, and long intervals between generations. Genetic evidence adds weight to losses occurring far apart. Available studies suggest extensive connectivity across the North Pacific rather than neatly isolated regional populations. If that picture is correct, declines observed near Taiwan and those detected farther north belong to the same broader biological system. Geographic distance does not guarantee an untouched reservoir waiting elsewhere. A population spread across an ocean can still function as one connected inheritance. Management has not fully caught up with that reality. Across much of the Pacific, protections designed specifically around Pacific sleeper shark biology remain limited. In United States federal fisheries, the species has often been managed within broader shark groupings rather than through a framework tailored to its unusual growth, reproduction, depth distribution, and vulnerability. Catch records can count animals that reach fishing gear, but they cannot directly count the mature population living beyond routine sampling. Researchers have acknowledged that a large portion of this population remains unseen and unsampled. That sentence carries unusual weight because monitoring depends on what can be observed, while the most consequential animals may occupy the least observable habitat. Science has recognized the Pacific sleeper shark since 1944, yet more than 80 years later, much of the evidence still comes from younger animals entering accessible fishing depths. Meanwhile, signs of decline have appeared before basic adult biology has been resolved. The difficulty is no longer merely that knowledge is incomplete, it is that time is passing inside that incompleteness. An animal can disappear gradually from places where it once moved, leaving behind datasets detailed enough to record the decline, but never complete enough to show precisely what was lost. An ecosystem does not experience a predator as a name on a species list. It experiences the animal through countless small events. A fish removed here, a carcass consumed there, nutrients carried downward inside a moving body. Across the North Pacific, the Pacific sleeper shark participates in these exchanges over immense distances and depths. Its disappearance would therefore remove more than a large shark. It would subtract a collection of ecological processes that other organisms have developed around. Large predators often influence ecosystems far beyond the animals they directly consume. Their presence changes survival, distribution, competition, and feeding behavior among prey. On land, the return of wolves to Yellowstone helped alter herbivore behavior and vegetation patterns, with effects spreading through the landscape. Marine systems reveal comparable complexity when shark populations decline. Changes among predators and prey can reorganize fish communities, sometimes reaching organisms several feeding levels below the original loss. Such cascades are difficult to predict because food webs behave as networks rather than simple chains. The sleeper shark occupies a broad predatory position within that network. Its known diet includes fishes, giant Pacific octopuses, and marine mammals, placing pressure on several very different groups at once. Predation does not necessarily keep every prey population low. Instead, it can help prevent particular species or size classes from becoming disproportionately abundant. Without that pressure, some prey could increase, intensifying competition for their own food and transferring the disturbance farther downward. The precise outcome in the deep Pacific remains uncertain, which is itself part of the risk. Removing a poorly studied predator creates consequences that may become visible only after the original cause has faded. Its role also continues after other animals die. Organic material descending from upper waters is precious in the deep ocean, where food arrives irregularly. Dead fishes, marine mammals, and other carcasses carry concentrated packages of energy toward depth. Sleeper sharks can consume this carrion, breaking large deposits of tissue into metabolism, waste, and smaller material available to other organisms. Around a whale fall, every consumer participates in transforming a single carcass into nutrients that can circulate through a deep community for years. A large scavenging shark becomes part of that redistribution. Movement adds another dimension. Pacific sleeper sharks can occupy shallower waters and descend far deeper, carrying material acquired in one layer into another. Food consumed hundreds of feet below the surface can later be metabolized thousands of feet down. Through respiration, excretion, digestion, and eventually death, the shark relocates carbon and nutrients through the water column. Multiplied across many individuals and long lifespans, these movements become threads within the biological transport of organic matter into deeper ecosystems. No obvious substitute waits to assume all of these functions. Other deep predators hunt fishes. Other scavengers process carcasses. Other animals migrate vertically. Yet ecological replacement requires more than performing one similar act. The Pacific sleeper shark combines enormous body size, broad feeding habits, deep habitat use, scavenging ability, and movement across layers of the North Pacific. No known species duplicates that complete ecological profile across the same range. This is why extinction cannot be measured only by counting what disappears. A species is also what it does. The Pacific sleeper shark links living prey, fallen carcasses, shallow production, and deep communities through the ordinary acts of feeding and moving. Those relationships have been woven into the Pacific over evolutionary time. Remove the shark, and the ocean does not simply contain one fewer animal. It loses a predator, a scavenger, a carrier of nutrients, and an ancient pathway through which energy has traveled from one depth to another for generations. Human admiration has always favored predators that announce their abilities. A great white shark rises from the sea beneath its prey. A killer whale turns cooperation into coordinated pursuit. A tiger compresses muscle into a sudden charge. These moments fit easily inside human ideas of mastery because they are visible, rapid, and unmistakable. They transform predation into spectacle, giving the eye a beginning, an acceleration, and an ending. The Pacific sleeper shark offers almost none of that. Its success unfolds without the movements that usually define a celebrated hunter. There is no airborne breach, no prolonged sprint, no obvious display of force before contact. Its body advances with extraordinary economy, gathering information while spending as little energy as possible. Yet harbor seals, sea lions, salmon, squid, and other capable animals have appeared among its prey. The outcome resembles that of far more energetic predators, even though the method belongs to another physical world. Our definition of an exceptional hunter was built largely from animals available to observation. Cameras favor open water, daylight, speed, and encounters that unfold within minutes. Human vision favors the same things. From that narrow window came an intuitive rule that successful predation should look powerful while it happens. The deep Pacific has no obligation to follow that rule. Much of Earth's habitable ocean exists beyond useful sunlight, under immense pressure, at temperatures where conserving energy can matter more than producing bursts of speed. Here, efficiency changes the meaning of perfection. A predator gains little by racing through cold water if acceleration consumes more energy than the next meal can reliably replace. It gains more by approaching without creating strong disturbances, maintaining sensory awareness, and remaining prepared for an opportunity whose arrival cannot be scheduled. The sleeper shark's low metabolic demands allow readiness to continue without the expense associated with constant pursuit. Waiting, in this setting, is not inactivity. It is a sustained predatory condition. Every moment of that waiting contains work. Receptors remain available to minute electrical signals produced by living tissue. The lateral line registers disturbances passing through surrounding water. Chemical information arrives with currents. Muscles maintain direction and position while movement remains restrained. Instead of repeatedly switching between rest and explosive pursuit, the shark can inhabit a prolonged middle state, conserving energy while remaining receptive to whatever enters its sensory field. Patience becomes physiology expressed as behavior. Human time makes such a strategy difficult to appreciate. We naturally isolate the encounter, measuring seconds before contact and minutes spent feeding. Evolution keeps a different account. It records whether enough meals are obtained across years, whether enough offspring survive across generations, and whether a body plan remains workable as oceans change around it. Under that accounting, spectacular victories have no special value. Persistence is the result that remains. Across deep time, many marine predators have appeared with formidable bodies and specialized ways of hunting. Their lineages eventually ended. Sleeper sharks endured through enormous changes in climate, prey communities, coastlines, and competing species. Their survival does not prove that slowness is universally superior. It reveals something subtler. Success belongs to the strategy that fits its environment closely enough to keep working when conditions punish unnecessary expense. The ocean's definition of perfection, therefore, looks different from ours. It has no requirement for spectacle, aggression, or speed. In cold water beneath the reach of sunlight, perfection can be restrained, carried almost beyond human intuition. The Pacific sleeper shark embodies that possibility in flesh, moving only as much as necessary, sensing what cannot hide, spending little, and allowing time itself to become part of the hunt. Its achievement is not that it dominates every encounter. It is that, generation after generation, it has remained. The first mistake was not made in the deep ocean. It was made at the surface, in the language and expectations humans carried with them. A shark brought upward appeared sluggish, so its family received a name associated with sleep. An animal encountered around carcasses was treated largely as a scavenger. A species rarely observed became a species poorly studied. Again and again, absence from human view was allowed to resemble absence of complexity. That pattern reveals more about observation than about the shark. Human understanding begins with senses shaped for daylight, air, and nearby objects. Scientific institutions are more sophisticated, but they still inherit practical versions of the same limitation. Animals that surface, migrate predictably, gather in recognizable places, or perform behaviors cameras can capture are easier to study. Great white sharks, whales, and dolphins can become recognizable individuals, documentary subjects, conservation symbols, and long-term research programs. Their visibility creates opportunities for attention, and attention attracts funding. The Pacific sleeper shark occupies the opposite condition. Its life takes place largely beyond ordinary observation, without regular appearances that can sustain public familiarity. It offers few photographs, fewer prolonged encounters, and almost no behavior that researchers can watch repeatedly in natural conditions. That invisibility has no relationship to ecological importance, yet the practical consequences are substantial. What cannot be observed easily is expensive to investigate. What is expensive to investigate often remains poorly described. What remains poorly described can struggle to compete for conservation resources against species whose lives are already legible to us. For decades, Data Deficient captured that problem in 2 words. The classification did not mean the shark was secure. It meant available information could not support a confident assessment of extinction risk. When the International Union for Conservation of Nature reassessed the species as near threatened in 2021, the change carried an uncomfortable implication. Evidence of decline had become visible before the animal itself had become well understood. Uncertainty and deterioration had occupied the same years. That sequence matters beyond one shark. The deep ocean contains thousands of species whose abundance, reproduction, movements, ecological relationships, and sensitivity to disturbance remain incompletely known. Many receive little attention until fishing, mining interest, warming, pollution, or another human pressure creates an immediate reason to measure them. Knowledge then begins after influence has already arrived. The scientific deficit is therefore not distributed randomly across life. It accumulates around organisms living far from observation and far from obvious economic value. The sleeper shark gives that deficit a recognizable body. Its history shows how easily unfamiliar biology can be interpreted through familiar standards. Slowness looked like weakness because speed is conspicuous. Scavenging looked primitive because active predation was difficult to witness. Rarity of encounters suggested rarity of animals because inaccessible abundance is hard to imagine. Each assumption became reasonable only when the human viewpoint was mistaken for a complete one. For most of its evolutionary history, none of this mattered to the shark. Human recognition was irrelevant to whether it found prey, reproduced, or moved through the Pacific. Understanding became necessary only when industrial activity acquired the ability to reach the same depths. Hooks, nets, vessels, and changing ocean conditions can affect an animal without first revealing how its life works. That creates a distinctly human responsibility. The Pacific sleeper shark does not require admiration, mythology, or even familiarity. It requires decisions informed by biology rather than visibility. Millions of years produced an animal fitted to a world humans entered only recently. Our knowledge arrived later still. The mirror it holds before us is simple. Ignorance does not prevent impact, and what remains unseen can still be lost. The next decisive evidence may come from something smaller than a coin, the layered lens inside a sleeper shark's eye. Unlike many tissues, the central proteins of a shark's eye lens are formed early and remain metabolically stable. Researchers can analyze radiocarbon preserved there, using the chemical history of the ocean as a clock. The National Oceanic and Atmospheric Administration has been collecting lenses from Pacific sleeper sharks taken as bycatch, building a path toward one of the species' most important missing measurements: reliable age. Confirmed ages would change far more than a line in a field guide. Growth rate, age at maturity, generation length, and natural mortality all feed into population models. Without them, estimates of recovery remain uncertain. With them, scientists could begin distinguishing a population capable of replacing losses over decades from one whose biological schedule unfolds across much longer spans. The same tissue that once helped an animal perceive its surroundings may therefore become a record of the tempo of its entire life. Movement presents another frontier. New acoustic tagging systems are being designed to function at depths beyond 3,000 feet, where conventional tracking becomes increasingly difficult. Tags placed on large, mature sleeper sharks could reveal whether adults follow recurring corridors, gather in particular regions, descend far beyond the depths represented in fisheries records, or travel toward specific breeding grounds. At present, those possibilities remain largely hidden because most tracking has involved few animals and limited periods. Observation must accompany those signals. Remotely operated vehicles can remain at depth while cameras, sensors, and sampling equipment search habitats without hauling animals upward. Used systematically in regions where mature sleeper sharks are suspected to occur, these vehicles could transform isolated encounters into sustained biological records. Courtship, mating, interactions between adults, responses to prey, and associations with particular seafloor features have never been adequately documented in the wild. A carefully planned observation program could replace inference with behavior seen directly. Genetics offers a different map. Existing evidence suggests broad connectivity across the Pacific, but the available samples represent only fragments of an enormous range. Tissue gathered from Japan, Russia, Alaska, Canada, Mexico, and recently confirmed animals farther into Asian waters could test whether that apparent unity conceals smaller breeding groups. Such structure would matter immediately. A locally depleted population might not be replenished rapidly from distant waters, even if the species appears genetically connected at the scale of the basin. Scientific resolution, however, has value only when management can respond. Treating Pacific sleeper sharks as part of a broad shark complex obscures the very traits that make them vulnerable. Species-specific bycatch accounting, release protocols, mortality estimates, and limits informed by life history would allow regulation to reflect this animal rather than an administrative category. Because its range crosses national boundaries, those measures would also need coordination among governments and fisheries operating around the North Pacific. No single jurisdiction can protect a population that spends its life moving through waters divided only on human maps. None of these methods belongs to an imagined future. Radiocarbon analysis exists. Deep-rated tags are advancing. Remotely operated vehicles already work under immense pressure. Genetic sequencing can extract population history from a small tissue sample. The missing element is sustained deployment at the scale the species requires. The Pacific sleeper shark is therefore not beyond saving, nor permanently beyond understanding. Each dated lens can sharpen the clock of its life. Each mature animal carrying a tag can draw another line through an unseen geography. Each genetic sample can clarify how distant populations remain connected. The distance between uncertainty and knowledge can be crossed, piece by piece. Whether research crosses it before decline outruns discovery depends less on possibility than on priority. There is a strange mismatch between the clock carried by the Pacific sleeper shark and the clock governing the danger around it. Its lineage was shaped across millions of years, while industrial fishing has transformed the North Pacific within less than 150 years. On a geological scale, that interval is scarcely a mark. For an animal that may require around 50 years merely to reach maturity, however, several human generations can contain a biological crisis from which recovery may take far longer. 20,000 years ago, near the height of the last glacial maximum, the Pacific looked profoundly different. Sea level stood roughly 400 feet lower than today. Continental shelves were exposed, coastlines shifted outward, currents changed, and surface ecosystems reorganized beneath a colder climate. Sleeper shark ancestors remained within that changing ocean. As the great ice sheets later retreated and seawater rose across drowned shelves, the world above them transformed again. Their lineage continued through the transition, carrying an evolutionary inheritance built for persistence rather than rapid replacement. That inheritance gives an old individual another significance. A shark that has lived for centuries has occupied the same ocean through conditions that younger animals never experienced. Chemical signatures incorporated into tissues can preserve information about diet, water masses, and movement. Parasites can reveal connections among hosts and regions. Changes in growth and body condition may reflect shifts in prey availability. Properly interpreted, such evidence turns an animal into a biological archive containing fragments of environmental history written by metabolism rather than ink. If the oldest Pacific sleeper sharks reach ages approaching those suspected from their biology, some individuals alive now may have entered the ocean before industrial fleets acquired engines, sonar, enormous trawl nets, and fishing lines extending across miles of water. Their lives could overlap the transformation from comparatively localized harvest to extraction operating across entire marine regions. They may have passed through decades of changing salmon abundance, altered marine mammal populations, expanding fisheries, warming waters, and growing human influence without ever approaching the surface closely enough to become familiar to us. Longevity, though, is not immunity. The conditions emerging now combine pressures that do not resemble a single ancient climatic event. Surface waters are warming, marine heat waves are changing prey distributions, oxygen patterns are shifting in parts of the ocean, and food webs are responding unevenly. Industrial activity can reach deep habitats through fishing gear, pollution, and potentially expanded seabed extraction. Evolution has given sleeper sharks flexibility across enormous environmental change, but past survival cannot guarantee success against the speed and combination of modern disturbances. Their future, therefore, carries an unusual contrast. Left with sufficient habitat and enough mature animals to reproduce, the species may possess the biological machinery to persist through another transformed Pacific. Yet the same slow life history that served it across deep time makes rapid population loss difficult to reverse. A lineage capable of enduring geological upheaval can still be diminished within the brief administrative cycles of only a few years by which fisheries are assessed and regulated. The Pacific sleeper shark has passed through ice ages, shifting coastlines, reorganized food webs, and immense climatic transitions. What confronts it now is measured less by geology than by economics. Fishing rules determine how many accidental captures are tolerated. Research budgets determine how quickly uncertainty can become knowledge. International agreements determine whether protection follows an animal across borders it cannot perceive. Its endurance has always depended on having more time than the disturbances surrounding it. For perhaps the first time in its history, the amount of time available is being set by us. At the beginning, the Pacific sleeper shark seemed to contain a contradiction. A predator of enormous size moved through darkness with clouded, damaged eyes, yet somehow captured animals that appeared faster, sharper, and better equipped to escape. The contradiction fades once the deep Pacific is considered on its own terms. Down there, success does not require the tools that dominate the surface. It requires removing costs that no longer provide an advantage. Vision becomes less important where sunlight never arrives. Maintaining elaborate sensory machinery demands energy, and energy is precious in cold water where meals may be separated by long intervals. The sleeper shark does not experience its damaged sight as humans would. Its world is assembled through other channels. Electrical fields reveal living bodies. Pressure changes, trace movement through water. Chemical signals persist along currents. Together, these senses provide information suited to a place where an excellent eye would still have almost nothing to receive. Even the parasite associated with its corneas fits differently within that perspective. Damage to the eye appears devastating when judged by an animal whose life depends heavily on sight. For a sleeper shark living far below useful sunlight, impaired vision may matter far less than we instinctively assume. Evolution did not need to produce a substitute for daylight. It shaped an animal capable of functioning where daylight had ceased to matter. Its slowness deserves the same reconsideration. Speed is expensive. Muscles accelerating a large body consume energy rapidly, and pursuit makes sense only when prey can detect an attacker early enough to flee. The deep offers another arrangement. A predator that approaches with minimal disturbance can conserve energy while denying prey the warning needed to begin an escape. What looks sluggish beside a great white shark becomes exact when measured against the physics of its own habitat. The sleeper shark is not unfinished, it is calibrated. That calibration may have been refined across millions of years, yet much of the animal remains beyond direct observation. Even in 2025, scientists still cannot state its maximum lifespan with confidence. Reproduction has never been observed in the wild. The largest adults remain almost entirely absent from research collections. Their movements, mating grounds, and deepest regular habitats are uncertain. Population decline is difficult to measure because the portion of the species most important to reproduction is also the portion least accessible to study. A glimpse from the South China Sea captures that boundary between knowledge and absence. At a depth of roughly 5,345 feet, scientists lowered bait into water beyond ordinary human experience. A Pacific sleeper shark eventually appeared. It approached the carcass without spectacle, fed within the reach of cameras, and then departed the illuminated patch created by the expedition. For a short interval, technology made one individual visible. Beyond that artificial pool of light remained an ocean large enough to conceal almost everything else about its life. The shark itself carried none of our categories with it. It did not know that scientists debated its age, that conservation assessments described it as near threatened, or that a camera recorded evidence valuable precisely because such encounters are scarce. It responded only to the information its senses provided, following a biological routine older than the institutions trying to understand it. Then the animal moved away from the bait and descended beyond the camera's useful view. Nothing about that departure required drama. Its existence has never depended on being witnessed. Long before submersibles, fisheries, surveys, conservation lists, or deep ocean cameras, sleeper sharks were already moving through the Pacific according to rules written by pressure, cold, hunger, and time. Some things do not need to be seen to be real. The Pacific sleeper shark has always known this. It has built its entire existence around it.